Hello, everyone. It's December 23rd, 2020, the worst year ever. Tomorrow is my 32nd birthday, and today we filmed the last episode of the year for Lady Scientist podcast with Franziska Seeger. She's a computational protein designer and has had a really interesting career, including working on X-ray crystallography, which is basically the way that we're able to determine the structure of proteins. It's a fascinating field. It requires some intense dedication, maybe a little bit of magic. We talk about that in the episode. But halfway through, I found out that she played chess competitively from a young age all the way through college and was basically internationally ranked. So I got super excited. I love the show, The Queen's Gambit, on Netflix with a bunch of great actors, and just love how well done the show is. And so we get into some of her experience in real life of playing as a woman, as a young girl in these circuits, and the intensity of those situations. So thank you all for listening, for subscribing. Comment with suggestions, questions. If you know people who you think would be great on the show, I'd love to hear about it. All of our links are available on the website, ladyscientistpodcast.com, which my wonderful husband built and made, and it's looking beautiful. So thank you for joining us on this journey in this crazy year, 2020. I know it's been kind of a shit show, but this has been such a great outlet, and I'm hoping that you are all getting so much inspiration, knowledge, positivity from the experiences of our amazing guests. So thank you.
So normally I ask before we start recording what, like, to make sure that I have your job title down
Yeah.
exactly, but I'd love for you to just tell us what your
Yeah.
job is and how you would describe yourself as a scientist.
Okay, so my official job title is kind of long. I'm a senior computational protein design scientist at Novo Nordisk here in Seattle. That's kind of a mouthful. I don't know why they gave me such a long title. Senior scientist would have been just fine, you know. But I like it, and it's kind of very descriptive for what I do. Which is, I've been at Novo Nordisk for 3 months now, and I'm working as a computational protein design scientist or engineer. I'm primarily working on rare diseases. And what I do there is kind of collaborate with teams of biologists or people who are kind of like testing very early drug candidates, kind of like in early development. And I evaluate targets for the feasibility for the computational design of small mini proteins or peptides, specifically targeted at oral availability.
Amazing. So you've only been at Novo for 3 months?
Mm-hmm, yeah,
and
exactly.
You were at Amazon before that?
Exactly, yeah. I was at Amazon for almost a year and a half before that. And before that, I was a postdoc in the Baker Lab.
In the Baker Lab. Yeah, I was gonna say the person I had on the last episode, it's kind of, I think, similar to you and I. Like, we've met once. Have we met twice, maybe, at like a Women in Bio event?
Yeah, there was a Women in Bio event at Lyle that I remember, where I think Brian introduced us, or we ran into each other, but like we had the Brian connection.
Yeah, we have like similar circles in Seattle, but we've never like hung out or anything. So
exactly,
it's kind of a fun, like, you have this parallel path to me in a way, and so I'm excited to like get into some of that, because I don't fully know like what you've worked on before, what you're doing now, so it'll be fun to like get into that.
That's awesome.
So can you talk, like, what was your transition from Amazon to Novo? Like, what was that?
Yeah, it was kind of weird to start a job in a global pandemic, to be honest. Like, I've been to the Novo office a couple of times, mostly for IT reasons, but I've never actually worked there. The transition was kind of interesting because it's very different to work for a tech company and then transition into a Danish pharma company. And so I had to learn a lot about like the cultural differences of Amazon versus Novo, but it's been actually fairly easy because Danish people are incredibly nice. And everybody is super friendly, and I had never really worked for a pharma company before, and so what I really appreciate is kind of that there are so many experts for every single detail of the process. So when I was a postdoc, I would, design my, identify the target that I wanted to engage, and then design my potential binders against that target. And I would have to test them, order them myself and test them myself, and then analyze the data that I got and then iterate on them. But at Novo, everybody kind of has their own speciality, and I can just really focus on what I'm best at, which is computational protein design. And that's been really fun.
First led you to protein design, and kind of how you got into it?
Yeah, I think that's kind of the path of how I became a scientist, or how I got to do what I'm doing right now. So it kind of all started, I'll start at the beginning. I've always been really fascinated with science, and in my teenage years, I was really fascinated with physics because my uncle is a physicist, and my mom was diagnosed with multiple sclerosis when I was fairly young, when I was 10, and I tried to read about it, and I learned that at that time, which is still true today, we really don't understand that much about the molecular mechanisms of disease. But what I did learn is that proteins, through reading, but also through my high school biology classes, that proteins execute essentially all important functions in the human body and in the cells. And they are kind of the executors of disease. And when I studied biochemistry as an undergrad, my mind was just completely blown by, I distinctly remember learning about cloning proteins into, other organisms for heterologous expression. I'm just whoa, this is the coolest thing. So I decided to pursue a PhD in protein crystallography and structural biology because I'm a very visual person. I've played competitive chess for most of my teenage years. So this kind of pattern matching and three-dimensional visualization really spoke to me. And yeah, I pursued a PhD in structural biology on a drug target that was highly implicated in cardiovascular diseases. The enzyme is called soluble guanylate cyclase, and I've had a love-hate relationship with it. I loved it and it hated me.
Relationship. I love that.
I really liked it, but it didn't like me back that much. So it was really fascinating, and I learned a lot about protein-protein interactions and how they are really important in mediating health and disease, because this protein itself consisted of two different chains that needed to kind of come together in exactly the right way in order to make a productive product. And that was so fascinating to me that shifts of a couple of angstroms can really make it the difference between pulmonary hypertension in a healthy individual. And I decided that I wanted to go further down the protein-protein interaction path. And at the same time, I also recognized that my partner at the time was a computer scientist. And so I kind of had this peripheral exposure to data science and machine learning and all of that kind of stuff. And so my appreciation for protein-protein interactions and kind of this fascination for this upcoming field of data science and machine learning kind of came at the same time. And then I learned about David Baker's lab in Seattle, and I was just oh wow, I really want to postdoc with this person, because it seems they are working on designing protein-protein interactions from scratch to interact with specific disease states, but they're also using computational tools to achieve that. And I thought that was the coolest thing, and I really wanted to learn more about that. And so yeah, I interviewed with David, and he offered me a postdoc position, and I moved to Seattle from the Bay Area where I was at. I was at Lawrence Berkeley lab. And and when I came to David's lab, I realized that the protein design program Rosetta is not everything that I had hoped it to be. It's, it can do certain things really well, but other things it can't do so well, and it requires a lot of manual input and kind of like curation in order to really make projects work. And I realized that this vision of making completely new proteins from scratch to engage any drug target of interest was kind of music of the future, as we say in German. And but I was really fascinated by this idea that you could theoretically just take any structurally defined drug target and make a custom protein for it, to interact with it in like the specific way that you needed for whatever indication you were interested in. And so I embarked on this giant project, which we called the protein-protein interaction benchmark, to test all different protocols that we had to generate completely de novo protein-protein interfaces from scratch. And I specifically targeted, my specific focus was still auto-inflammatory cytokines, because I was kind of always fascinated by that, starting from when my mom was diagnosed with multiple sclerosis, and like went a little bit away from that with cardiovascular diseases, even though there's a lot of inflammation going on there too. And also worked with many other people in the Baker lab to kind of like really have a plethora of different targets to really make this method generalize. And yeah, I designed kind of the first de novo binder against the IL-17 and IL-23 receptor cytokine. And I believe that is currently being commercialized out of the Baker lab by former colleagues of mine. And yeah, that was super cool to learn the ropes of computational protein design. I took a lot of data science classes while I was a postdoc in order to be more efficient programming and to really be able to analyze these giant data sets that come out of Rosetta. And yeah, that was just overall a pretty cool experience, and kind of like my transition from being a wet lab scientist to a computational scientist. Yeah, and then, yeah, that's kind of how I got to protein design. And then I was always a little bit nervous as like an international person in the US about what my career would look like and what my dependency on my visa status would be. And so I treated my career search, or my career design, almost as like a research project to figure out what exactly it is that I wanted to do after I finished my postdoc, because it was always pretty clear to me that I wasn't really interested in an academic career, much to David Baker's disappointment. We've had some heated discussions about this.
Oh, I'm sure.
It's kind of sad that in most academic circles, like alternative careers are not really respected as much as becoming a professor. But I really, just knowing what you don't want isn't necessarily enough to pursue what you do want. And I was really lucky that through a lot of networking at Women in Bio, I kind of very serendipitously got my first consulting gig about 2 years before I finished my postdoc. I was put in touch with somebody who had just started a startup in San Francisco and was hiring a data scientist. And I was really interested in how companies are looking at the intersection of data science and protein engineering. And yeah, that really gave me some initial insight into what it's like to work for a company. And I had this consulting company for the last 2 years of my postdoc. And yeah, it was super interesting. Amazon was actually one of my clients, and they wanted me to come on board full time. And I was kind of evaluating whether I would want to spin out my own company out of the Baker lab, or, join a larger, more established company full time right out of my postdoc. And eventually I decided that it would be a really cool opportunity to join Amazon and kind of experience what that would be like. So I did that right after my postdoc as a research scientist at Amazon, working on the intersection of like life sciences and machine learning, which was really fascinating just from a learning perspective, and also completely different environment to academia, which is like all I had known apart from my consulting work. And then I decided to join Novo just like for the sheer opportunity of like working for a company that can really push molecules through the clinic and have like a real impact.
Wow. You touched on the kind of cultural shift from academia into Amazon. Can you talk about that a little bit more and what that, big tech environment is like?
Yeah, it was kind of interesting. I think we were in like a little bit of an Amazon bubble, since we weren't necessarily involved with like any of the daily cloud or retail operations, obviously. What I really enjoyed at Amazon was this clear shift from being in an individual contributor role and interacting with other people who also had their primary objective of having a first author paper in whatever field to achieve their next career stage, to really being on a team and having a joint goal that we could work towards. And it didn't really matter who contributed what, as long as everybody contributed to the goal and we got it done in the end. And I think that's just much more in alignment with my personality and how I operate, because like I just really want to see things work and like do cool science, but I don't necessarily care whether I have like 80% of the contribution to this project. I think if somebody else can contribute to this project and we can all work in a team and we can leverage our individual strength, then that's much better than me trying to take a long time to do something that's not necessarily in my wheelhouse.
Absolutely.
And that's been super cool. And just like the environment at Amazon that I was in was very startup-y, I would say. And it was pretty cool to just ideate new ideas and come up with different projects that kind of like fit the unique capabilities of Amazon, which was kind of like a completely new set of parameters that I was used to before.
Sure. Wow. And were you working like, because I know that's how the idea of how Amazon is run is that each team is supposed to act like a little startup within this larger organization.
Mm-hmm.
Were you, what building were you in? What was your team's composition like? Because I know a few scientists who have gone to Amazon now, and I'm always curious like what that looks like. Do you guys have like a shared office, or are there wet benches at all? I mean, you do dry computational biology now, but
yeah I have to be a little bit careful kind of like to answer these questions, obviously.
No worries.
We were
broad strokes.
The team composition was really interesting. I exclusively worked with PhD level scientists of like all sorts of different domains, which was really, really cool. My manager has a PhD, or like still has a PhD, but it's my former manager, so had a PhD in physics and was a machine learning expert. And there was another former Baker Lab colleague on my team who had a computer science and software development background from Microsoft, and there was another structural biologist with an MD background. So it was kind of and a medical doctor. It was just like really interesting to have like this type of team composition and to really shift. I would say I was like the most biologist person on that team, which is not something that I usually identify with. I feel like anything that is bigger than a protein kind of scares me, like cell level interactions, that's kind of like too much for me. So I don't self-identify as a biologist, but like I just like because I was the most wet lab trained and biology minded person on that team, like a lot of like these questions fell to me, which was really interesting. We were kind of a little startup within kind of like a larger undisclosed organization, and it was kind of cool and a little bit isolating too, because it was definitely not what we worked on was definitely not kind of like in the main wheelhouse of Amazon. So we were always kind of like the odd scientists in the corner a little bit.
Funny.
And yeah, it was really cool. It was kind of interesting to observe how a company structure works that was set up to be optimized for something completely different. But I really appreciated the style of, so Amazon has these leadership principles and also principles to approach problems, and one of them is working backwards. And you always have to work backwards from the customer and kind of like the problem that the customer has. And I think that a lot of academic grant writing would probably be a lot more effective if, you know, like we were trying to always argue from like a perspective of like solving a specific problem, or at least that's like what I think, because I'm like a very application-minded person. And the grant writing process was also completely different. Amazon operates in what they call PR FAQs, which are press release frequently asked questions. And the way that you propose new projects is to essentially write a press release about, the press release that would be published if your project is completed successfully, which is kind of cool and was a new way for me to think about science.
Huh. I had not heard of that before. That's so interesting.
Yeah, it's
press release. Cool. What was the term again?
Frequently asked questions, PRFAQ.
That's so neat. I love that. Yeah, I think it's interesting. Like some Lyle, for instance, you know, we've thought a lot about culture and principles, and I think being close to Amazon, you can't help but hear about how these principles guide the work there. And it's interesting to like hear a scientist's perspective of like how you adapt a customer facing organization and principle structure to the science. I think that's pretty neat that you actually took something away from that and it guided your way of doing your work, which is in my mind so different from what most of the folks who work at Amazon here in Seattle at least are focused on doing, right? Which is like absolutely improving a customer experience.
Yeah, exactly. And like, I mean, sometimes, don't get me wrong, sometimes that was a struggle and there was definitely a disconnect between, you know, the scientific process and like how Amazon operates. But for me personally, it was just a really interesting learning experience. And while these leadership principles are really ambiguous and can be interpreted very individually towards everybody's individual liking sometimes, it is quite interesting how strongly they influence the company culture and how top of mind they are and how often they are used to argue for specific directions or to make decisions.
Interesting. So I know we can't get into the details, but were you still working on de novo designed proteins at Amazon? Was that the focus?
So I was doing protein design, but yeah, I can't talk about whether I was de novo or anything else. Yeah.
The reason I ask is because you mentioned, you know, after you had kind of gotten your footing in the Baker lab, that you realized that de novo proteins in some sense are the music of the future. I like that term. And I think I've also had some realizations after working with some of those folks, just to the limitations and also things like we don't know necessarily. Or I do want to ask you this other question. Sorry, I'm all over the place this morning.
No, this is great.
But have you had an arc of like you were doing de novo proteins for a while and now you're back to more of like rational protein design or borrowing proteins from nature, you know, just curious if you've like shifted back at all or if you're still mainly using Rosetta and designing de novo proteins?
I think that's a really great question. So I think the way that I'm framing this in my head is I've really done technology development in the Baker lab that is not necessarily ready for prime time yet, if that makes sense. So I think in the Baker lab, I was really driven by this vision of developing algorithms that one day could just like be turnkey and automated to really generate what we call binders, just like de novo proteins that bind target of interest in like a specific fashion that is needed for functionality. And while I think that was really cool and kind of like an ambitious goal and worked at like a very low percentage success rate. Oh, I think my neighbors are having their
It's okay.
lawn mowed right now. I hope that's not too loud.
No, that's fine.
While, you know, I think that was a really fun academic exercise, and I think it's definitely gonna be important in the future if you're actually trying to develop new therapeutics or vaccines or diagnostics. It's much smarter to leverage the biological or like native protein information that you have. So in reality, if you're trying to work on real world applications in whatever shape or form, you might be well advised to incorporate some of the information that you have. So to speak in a little bit less abstract terms, like very often, or if we're lucky, we have crystal structures of how our protein target of interest engages with a native protein in the body or something like this. And so borrowing information of how other proteins interact with the target of interest and maybe taking hotspot residues from that protein or even full secondary structure elements to use as inspiration to make smaller mini protein binders or peptides is generally not a bad idea, because then you have a starting point that is a little bit more reliable. And you're not kind of necessarily dependent on a really well-working de novo interface design protocol, depending on like the shape and surface composition of your target that you want to bind to. That can be very challenging. And sometimes there was like a recent really cool preprint from the Baker lab that was incorporating something that I've been interested in a long time, which is used for antigen stabilization and development. So like having like discontinuous elements of like for instance, like a 3-dimensional B cell epitope, and trying to figure out how you can stabilize that into like a smaller manufacturable unit that you can then use as like an immunogen for vaccine development or something like this is super interesting. And so for these types of applications, you also don't really want to go completely de novo, because you need to elicit an immune response against like this natively occurring protein.
I see. Are there instances of these de novo proteins in clinical trials? That's like one space I'm not familiar with, and I'm just curious.
Yeah, I think the most the two examples that I can think of, and I have to admit that I'm not completely informed of like which stage they're at right now, but there are two companies that were founded by former people at the Institute for Protein Design. One of them is called PVP, Protein versus Protein, which manufactured an enzyme that can be formulated into a pill to degrade gluten in the stomach for celiac patients. And so that is currently, I'm pretty sure an IND has been filed. I don't know which clinical trial stage it's in, but that's something that where a native enzyme was stabilized and redesigned using Rosetta that is currently being pushed into the clinic for celiac's indication. And then something that's really cool that was done or pioneered primarily by my friend Danielle, who was my mentor when I first joined the Baker lab, is an IL-2 mimetic technology that is currently being pursued by a company called Neolukin that spun out of the Baker lab. And I know that they definitely filed an IND and might have entered phase 1, but I'm not entirely sure. And so everybody in the community is carefully watching specifically the IL-2 mimetic, to see, what the immunogenicity is going to be like and how the efficacy and how it just fares in the clinic, because there's just really very little information about completely de novo designed proteins from clinical trials. Like this is kind of like the bleeding edge of this technology.
Yeah, that's one thing that, I work in more of like the gene therapy cell therapy space, and so many of these technologies are just cutting edge and we don't have a ton of clinical data on them yet. And so it's interesting that, hopefully like in the next few years we'll have a better understanding of how the human body reacts to these reagents being present. So,
yeah, that's really cool. Okay, can I ask you a question?
Yeah, of course.
So I'm curious how you see, since I know you've worked with a lot of protein designers at Lyell, how do you kind of like see the future of protein design paired with like gene and cell therapy? Like some people that I'm talking to think that like cell therapy is the music of the future, and like protein design or protein engineering will just aid to make the appropriate switches or surface receptors, or like what you want to call it. So like I'm just curious how you see how these two fields kind of will merge in the future, and what's going to be the next cool thing there.
Yeah, I mean, in broad strokes, what I was working on was trying to merge chimeric antigen receptor T-cell therapy with some of these de novo proteins that have come out of the Baker lab. And I'll say like CARs in general are, these massive fusion proteins that have a binder, and often it's a SCFV, and then there's just all these different protein domains that have been mushed together to form this thing. And I think for me coming into that field, I was pretty impressed that they work so well at treating some of these hematological malignancies. And we still don't fully understand why they don't work great for solid tumor cancers. But I actually think the last couple years, the clinical trials that are coming out for things like melanoma with the BCMA CAR looks pretty promising. And I don't know, it's kind of wild to me that you can take a cancer patient who's been treated with every type of chemo and drug available, and they've failed and failed and failed at all of these different treatments, and they're really like the end stage disease, and you can take their T cells out of their body, transform them to be expressing these CARs, and put them back in, and their own T cells are going after the cancer and killing it. I just think it's kind of mind-blowing, right?
It's mind-blowing. Yeah, no, it's like it's super cool. I'm really impressed by what you're doing. So this is awesome.
Well, and I'm gonna lose my train of thought, but you touched on the protein design kind of advancing with this, and I think there's definitely room for improving the safety of these cellular therapies, because you've studied cytokines, so you know how hazardous they can be in the body. I mean, cytokines are responsible for a lot of the disorders like you were talking about with multiple sclerosis, and there are certain cytokines that are extremely deadly to other cell types and can really wreak havoc. So I think kind of dialing in the T cell product so that it's potent in the cancer tissue you're going after, but not normal tissue, is a really, really important part of those programs, and a lot of good people are working on that. But yeah, I don't know, like, I think there's a lot of exciting tools like the heterodimers that Zibo worked on, and the switches that Mark LaJoy worked on or developed. I think there's a lot of potential there, but I do worry about the immunogenicity question, and that with the CRISPR field also we're worried about that as well.
Yeah.
Because we just don't know. We don't have enough clinical data yet to say for sure that this expressing this giant enzyme that's not a human enzyme
Yeah, I know.
is gonna be like, okay. So, I guess time will tell, and we have to appreciate all the people who volunteer for some of these early trials, because it's
Oh,
really
absolutely. Yeah.
sorry, I got on like a tangent there.
No, that's fine.
Peptides, from what I remember of my experience with Novo here, they're interested in these like mini peptide binders. Is that correct?
Yeah, that's correct. So Novo has kind of like been historically a protein engineering and peptide company. So that's really like their identity. They've actually started a company and fared very well for a long time around a single molecule, and they're just recognizing that maybe they should branch out a little bit more, or at least that's kind of like how I'm understanding it after having been there for 3 months. And yeah, it's quite fascinating. I think there's quite a bit of interest around smaller peptides for metabolic diseases, and specifically like oral availability, and how things can be targeted to different tissues and different cell types, which is really interesting. And so, I mean, talking about cytokines again, like what I find fascinating, mind-blowing, and incredibly frustrating is that the same cytokine can have like completely different effects in different tissues, right? And there's concentration dependent effects, and it's really complicated how IL-17 can have like anti-inflammatory effects in like part of the body, and then inflammatory effects in other parts. And so I think trying to really figure out how to do tissue targeting and oral uptake to prevent the necessity of bi-monthly injections or something like this is really interesting.
How do you how is it like ligand based, the tissue targeting with these peptides?
Yeah, so I think we're just trying to figure this out, and I'm definitely not an expert on this, but what I'm understanding is that like there's this emerging data coming up that that is not necessarily kind of like how should I say this, like individual protein name based, but there's specific. There was recently like a really interesting GPCR paper that was published where which in my unprofessional opinion in this field was the authors presented data for the first time of how different isoforms of the same protein have different effects in different tissue types. And so I think this is kind of like the beginning of what I understand, and like maybe correct me if I'm wrong, is kind of like an underappreciated field of biochemistry, which is how do different isoforms of the same protein play wildly different roles in different tissue types. And so as like a structural biologist and biochemist, that's kind of complicated to me, because crystallographers kind of like to crystallize the dominant form and then pretend that the other isoform does something but like works essentially the same. And I know that's kind of like being a little bit mean to my field of origin, but that's definitely how I approached my PhD thesis. Even though there were other isoforms of the protein that I worked on, we really mostly ignored them. And I think there's recent data emerging that say that we probably shouldn't ignore all of these different isoforms and see what we can leverage for therapeutic applications.
Yeah, that's interesting. One of my early grad school studies, we were interested in how transcription factors determine the expression of different isoforms of a gene. And the main area that I refer to myself. There's one of the databases that was generated is like the tissue specificity of expression of these different isoforms of the gene, and it is pretty important to know when you're designing, say an RT-PCR assay that's going to measure the expression of this particular gene. You actually need to know which exons are in there.
Yeah.
Or else you might not be covering the correct, exon-exon junction. But there's so much that we don't know, right?
Yeah, so much. Yeah. That's why it's cool to do what we do, because we can always learn new things every day.
Yeah, that's awesome. Tell me, because I've worked with Matt, who was a crystallographer.
Yeah, Matt Big.
Yeah, and he's at Neolucan now. And I've heard that crystallography, it kind of has this reverence from other biologists that like it's a special kind of art form, basically. And I've even heard like that certain crystallographers, they only play like classical music when they're in the lab because there needs to be this, like perfect condition for the crystals to form. So can you talk about that? Did you have any like quirky crystallographers you worked with?
Oh yes, absolutely. It's an interesting field. I mean, there's definitely like a little bit of what I would call voodoo or black magic surrounding this field. I've definitely heard of crystallographers who only use like this very specific horsetail hair to start seeding their crystallization conditions, and they swear by like this one cat whisker or something like this. I mean, like, to be fair, like animal hair is used as kind of like a nucleation initiator for crystallization sometimes. So it is like an actual technique to mostly use a solution that has like formed maybe microcrystals to initiate seeding of larger crystals with like animal hair or other things that can like provide a nucleation basis or something like this. And but yeah, it's just, since we don't really know a priori what type of conditions a protein will crystallize in, if ever, everybody kind of like has their own secret sauce, and depending on what type of conditions have proven to be successful for you in the past, you kind of get a little bit attached to them. And so I would say that process it's systematic. I wouldn't necessarily say it's scientific.
I love that.
So my grad school protein was a heterodimer, and it was like and there was no covalent cross-link that would kind of like staple these two subunits together. So it was actually quite challenging to produce a predominantly heterodimeric solution. And I used a lot of natives mass spec to quantify how much homodimer of the two homodimer types do I have in solution, how much heterodimer do I actually have. And so my main contribution to this field as a graduate student was actually the crystal structure of a heterodimer of the catalytic subunit of the wild type human protein of this cardiovascular disease drug target, but I could never reproduce those crystal conditions. Even with the same protein prep and like the same crystallization buffers, all I got were heterodimeric homodimeric crystals instead of like the heterodimer crystal. But like the heterodimer crystal that I got crystallized to, or like diffracted to 1.9 angstrom resolution, and I got a beautiful data set. And there are, tens of thousands of refractions that point towards your model being correct. And so I had lots of philosophical discussions with friends during grad school whether this was science, because I couldn't reproduce what I had done, even though I had a giant data set that kind of like was consistent in itself. It's a little bit of like a dark art, and you can definitely, like crystallography is kind of like an interesting case when it comes to science reproducibility, because like nobody would probably argue with me that like the data point that I got in this crystal structure was not like an important scientific piece of data. And obviously like I published my work and like I deposited the structure because it was a contribution to the field. But other people who are talking a lot about reproducibility are saying like, well, it's not really science if you can't repeat it. But it really breaks down in like the field of crystallography a little bit.
That's interesting. I didn't know that. I mean, because that's what I hear. Like you're laying these trays, you have different buffer conditions. Again, I have no background in crystallography, and I really have a very loose understanding of how it all works. But you know, maybe there was like slightly different air pressure conditions that day or something that you just, you know,
Yeah, I
allowed.
mean, it's really, so you're operating with really small volumes, right? Like, so a lot of these trays are like, if you set up a robot tray, like you're operating with like, I don't know, 0.2 microliters or something like this. So, you know, we all know how precisely a human can pipette 0.2 microliters. Some better than others, but it's hard. And a lot of the crystallization buffers are highly viscous, you know, like there's a lot of glycerol or like PEG molecules. My favorite PEG molecule was PEG 3500 because that's how I got my crystal. But it's just like, it's so weird. This is like almost 10 years ago and I still remember this because it makes such an impression. If your solutions are not perfectly mixed to like a homogeneous solution, you will inadvertently diverge from whatever percentage of these viscous molecules you have on your tube. And so it's really hard to know exactly what the precise composition of your crystallization conditions are. And then, you know, as you know, like lab temperature changes. I did this in California, you know, like if the sun shines through the window, it's warmer than if it doesn't or something like this. It's just, yeah, that's a long-winded answer to saying like, we just have no idea how proteins crystallize and like,
Sure.
It's
Fascinating.
definitely black magic.
What was that day like though, when you realized that you had a heterodimer crystal?
Oh, it was awesome. You know, honestly, I doing a crystallography PhD is like a little bit risky. I feel like with you know, I feel like doing a PhD is hard work in any type of field, but I don't know about like many other areas of biology or biochemistry where your success is so binary. You know, like my goal was really to get a structure of this molecule, and my mentor and my advisor at the time like had this philosophy that like I should be focused on this one project and shouldn't have too many alternative projects. And I really put almost like all of my money on getting a structure like that. So it's like it was very binary success. So I was getting really worried. Like I think this was like well into my 4th year of my PhD, that like I just wouldn't be successful in achieving this goal, and then I would be left with kind of like a bunch of biochemical data that I might not really be able to publish well.
Wow.
So like I was
kind of terrifying.
Excited. Yeah, no, it's kind of terrifying, and also now I'm just like, you might have wanted to diversify your risk a little bit, but like,
yeah,
you know, it worked out.
Oh my gosh, that's crazy. So you mentioned that you played competitive chess growing up.
Yes.
Can you talk about that? Like,
yeah, absolutely. I when I was in 5th grade in Germany, my school and we had like I don't know how the school system works here in the US super well, but like we had like a specific head teacher for my specific class. And so he was the head of the chess club and also the youth, I don't know, the person in charge of like the youth in our local chess club. And I had learned the rules from my dad when I was really young and was always really fascinated by this game. And so I joined the school chess club, and with like a lot of support of my teacher at the time, I took chess training and coaching and played on like the what would be the state level in the US pretty quickly. And so it was super fun. Like I played in like national German competition, like individually and on the team level. And I actually played for like the women's Bundesliga in Germany for a while during college. So
wow,
I can talk about that for hours. It was definitely like a very formative period of my life, as I now know. I think only through years of therapy, I've really been able to unpack a lot of that.
wow.
It was an incredibly, I talked to somebody about this yesterday, but like I credit my ability to kind of like persevere in this heavily male dominated field of structural biology and computational protein design for this long, because I've just been exposed to these types of environments for a really long time. I can say that it was always, when I initially first started talking about it, after I haven't played competitively in about 10 years. And part of the reason why is that I experienced the environment to be incredibly toxic sometimes and not very warm and nurturing, unfortunately. I was able to, while I was in my teenage years, there were always kind of like peers my age and really good friends of mine who I could lean on. But in like the adult leagues and stuff, it's pretty heavily dominated by, unfortunately, humans with like not incredibly well-developed social skills. And while I could zone a lot of that out because I was just like so into the game and into the strategy and into improving, as I was getting older, it was kind of harder and harder to marry that with who I was becoming as like an individual. And so I stopped playing, unfortunately, but it was like definitely a super interesting formative time of my life.
Wow, that is so interesting. So obviously I'm going to ask if you've watched the Queen's Gambit.
I have. I feel like every single one of my friends is just like, oh, redhead chess prodigy. I wonder who should watch this Netflix series.
Oh my gosh.
I had wet palms for like the first couple of episodes. It was like incredibly, like, I don't want to say triggering, but like I definitely could feel the tension there. Wow. And like could really envision of like what it must have been like for her, you know.
Yeah, because she's a fictional character, right?
She's a fictional character, yeah, but like honestly a lot of like so this played a lot earlier, obviously, during the era, like actually I think this played during the era of the Cold War, if I'm correct.
Yeah, that sounds right.
Yeah. So obviously earlier than when I was born in 87, but a lot of like the treatment of women in this field rang true to me and kind of like mirrors my experience playing chess. I unfortunately made the experiments that like, you know, boys started crying when they lost against me, and like they shared with me openly that like the only reason why they didn't want to lose was because I was a girl, you know. And so
wow,
we're talking about 11, 12-year-old children who probably couldn't really explain to you what sexism is. So I think this just shows that some of these some of these biases are so deeply ingrained in our society, you know.
Yeah, absolutely.
And I had I think I was probably like 14 or 15, and chess is an incredibly objective sport. I call it a sport because you have like essentially a number associated with your name that represents your place strength, and that is calculated by an algorithm. So
okay, I'm gonna close this because it's bright, but keep talking.
Absolutely. Yeah, so you essentially have a number associated with your name that fairly accurately reflects on how strong you are as a player. And I've had like...
Can you share your score? Or
Yeah,
it changes over time? How does it work?
It changes, so it gets calculated in like and I actually don't really know, it's kind of a complicated algorithm that like I didn't really fully follow, but it gets calculated based on how you perform against players with like different other scores. So if you win against players who are stronger than you or have a higher number than you, then your number increases. If you lose against players who have a lower number than you, then your score decreases roughly. And so there's like a German national rating, and I honestly don't remember what it is, but there's also an international ELO rating. And I think when I was at my peak, I probably had an ELO rating of 2,150 or something like this, which is, you know, to give you like a frame of reference, like grandmasters have like 2,400 ratings or something like this.
Wow.
Yeah.
So you were up there.
I mean, I was not an international master or grandmaster, but like I was pretty good for like I was definitely playing like on the top of the field in Germany at my age group.
Wow.
Yeah.
And so like I'd love to hear more of your take on the show, because I read somewhere that some of the games, I don't know if all of them were, but they are actually based on real strategy, like real chess games.
Yeah, they like the chess checked out in that game. Like there was no fictional positions or anything. Like
wow.
It was, yeah, they really did
Was
well.
that neat to see, like having known, because even my husband, he's big into games and used to play chess, not like on your level, but he was saying as we're watching it, like, this is a real game, you know, like they're actually doing the moves and everything. So that was fascinating to me, who hasn't, you know, I was never a gamer. Like,
Yeah.
if that makes sense.
Yeah, no, that makes sense.
A reader growing up. So I actually felt like I learned a lot about the game just from watching that show, which is kind of incredible, right? Like,
yeah, it's super interesting. Like chess tournaments are just such a mind-blowing experience. It's like it's highly psychological, you know, like I think they like just the strategy of the moves, but then there's also kind of like how you interact and look at your opponent, you know, and how nervous you are and like what your body language is. And it's like the tension that is in the room. I remember that like my mom didn't come to any of my tournaments because she just couldn't bear the tension in the room.
wow.
And yeah, it's like it was incredibly well done, I would say. And I was really feeling for her during during the series. It was quite fascinating.
Wow. Yeah, they got that really well too. I like, you know, for some of her games where you see it on her face, like when she realizes that she's lost or she's won, and she has that, like, you know, towards the end, like that kind of sheer determination. And
Yeah.
I don't know.
It's addictive. It's like a very visceral response. Like it's quite addictive actually. I used to like play chess on my phone a little bit, and like I famously, like when I play chess, I really have no idea what's happening around me. Like people can say my name and I'm completely zoned out. So
wow.
it's kind of, it's interesting. Like I miss having like this laser focus and being that much in the zone. I know I'm capable of that. It's just really hard to recreate with other types of things. I can do that a little bit when I write code or like I look at protein structures, but it's like, it's kind of it sucks you into like this alternate reality where all you see is the board.
Wow.
Yeah.
That's fascinating. So one thing from the show that I actually really enjoyed was the Russian component of like her getting ready to play. Can you hear me okay? I'm just adjusting my
Yeah.
My ears are too small for my AirPods too, and so they'll like
I
slowly
have the
pop
same
out.
problem. I have the same problem. I feel like they should make better earplugs for small ears. I have tiny ears.
Yeah, I'm
Funny.
like tiny. Yeah, it's ridiculous. But was the Russian component, because I actually, one of my first mentors and long-term mentors is Russian, and you know, he trained me at the bench. He trained me how to pipette, and I was still in college at Berkeley actually. And at the time, you know, he was a little bit younger than he is now, obviously, and he was very intense. Like he definitely wanted me to be like laser focused on this goal of working on these therapies. And if I remember one time I was reading about the Olympics like in the break room in the paper.
Yeah.
And he criticized me for it. And I don't know, there's something about like that like Russian intensity in this like training environment that I saw reflected in the show, and it kind of like brought back a lot of those memories for me, which it's just an interesting way of like being brought up as a scientist, essentially, like being expected to like only focus on your science and like nothing else.
Yeah, no, it's like that really resonates with me. My chess coach growing up, like I had individual chess coaching lessons. He wasn't Russian, but he was Ukrainian, and I think some of the culture definitely translates. And it was just very regimented and very, you know, he was just like, you're really good, you just need to execute. And I think my criticism of like the chess culture is that humans aren't robots, you know, and you know, like you aren't a robot, and like just because you read about the Olympics in your break doesn't mean that you were any less dedicated to working on like these diseases that you were working on, you know. And so I think in my 20s and like maybe after my grad school, I think one of the most important lessons that I learned is that it's okay to be human. You know, you can't just operate as a robot all the time. That life really sucks if you're trying to be a robot. Because humans are just bad at being robots.
Yeah, exactly. That's so true. Yeah, we could get into like a whole other robot conversation. Like I feel like we could talk for hours, but you probably have things to do.
This was really lovely. Thank you so much for having me. This was super fun.
Yeah, it was super fun.